Hi, everyone. This is Pam Mace. And for those of you who don't know me, I am the Executive Director of FMDSA. And I would like to welcome everyone here this evening. We're very excited about this webinar, and I would really like to thank Drs. Olin and Kovacic, Jason, for being here today to share this exciting data with us. I've personally waited 24 years for this day, so I'm very excited. And we'd like to also thank all the researchers that participated in this and the patients. I've heard from quite a few patients. I was one of 83, so I think I need to get a T-shirt made that says, that. But one of 83 patients that participated that actually made this research possible. So without further ado, I think the majority of you know who Dr. Olin is. And we are going to start with Dr. Jeffrey Olin from Mount Sinai in New York. And for those of you who don't know, he has been with FMDSA since the very beginning. So over 2 decades with us. Dr. Olin?
Thank you, Pam. It's great to be here. I'm just paging through some of the people who are on. Hi, all my patients. So I'm going to give a 10, 15-minute overview of the clinical phenotypic manifestations of fibromuscular dysplasia. And then Jason's going to talk about the study that we just published, which really is an incredible study. You'll hear about it from Jason. So, as most of you know, fibromuscular dysplasia really comes in 2 forms. This is the most common form, the string of beads type. This is a kidney artery, and you see multiple areas of narrowing and dilatation. There were 2 major papers written, one in 2014 and another in 2019, which really summarized the state of the science at those particular times. And the last paper was the International Consensus Document on Fibromuscular Dysplasia. And even since that's been published, there's been a lot of new information regarding this condition. So just to summarize, fibromuscular dysplasia can have the string of beads, as you see here. It can have a very focal area of narrowing. It can have tortuosity of the carotid artery. This should be straight here. It can have dissections or tears in the artery. This is a carotid artery, and this is a coronary artery. And then it can have aneurysms. So those are the many faces of fibromuscular dysplasia. This is a carotid artery, and it shows the typical beating. And then the artery becomes normal again. And we don't know why this occurs in this part of the artery, but not other parts of the artery. But that's one of the puzzles that we need to figure out. Now, I show this because this patient, who I still take care of, came to me in around 2010, and she brought with her films, the kind of films you used to put up in an X-ray box. We didn't have any X-ray boxes to put the films up on, but there were angiograms of her renal arteries from 1975. And they looked at carotid arteries and renal arteries. They looked exactly what you're seeing here. In other words, in all of those years, there's been no progression of disease, and there's been very few reports, if any, of the presence of beating in an artery where it was not present the first time that you did imaging on the patient. So some patients with fibromuscular dysplasia end up getting a balloon angioplasty. This type of fibromuscular dysplasia, though, occurs on the average in predominantly women in their 50s. And many of them have had high blood pressure for so many years that the angioplasty no longer is worth doing. But if you've had high blood pressure for a short period of time, this could be a way of curing the high blood pressure. This kind of FMD is focal FMD. This is a focal narrowing. This is a tubular narrowing. And This type of FMD usually has the sudden onset of very high blood pressure. And if you do an angioplasty on this, the blood pressure is often cured and they require no blood pressure medications. So I like to show this because many of you think FMD is an incurable disease. Well, it's incurable for many people, but for this patient, it was curable. She said, "Thank you for curing my FMD." She had this very severe narrowing of a branch renal artery causing blood pressures over 200. And she sent me this lovely vase as a thank you present. So in order to call it FMD, you have to have either the string of beads or that focal area of narrowing. These other things, dissection, ectasia, that's an enlargement of an artery, aneurysm, or tortuosity, the curviness, they're often present in FMD patients. But if you only have these four things, you can't call it FMD. Now, I think we're missing a lot of FMD because let me just show you an example of something we've seen multiple times. These are three sisters. One sister, sister 3, had FMD of the internal carotid arteries and SCAD, spontaneous dissection of the coronary artery, and an aneurysm at the celiac artery. The second sister had FMD of the internal carotid artery, the renal arteries, and the right internal and external iliac arteries. And the patient that I saw first only had a splenic artery aneurysm and arterial tortuosity. Now, are you telling me that this patient doesn't have FMD? Of course they have it, but she doesn't have the string of beads, so we can't really call her FMD. And if you don't have other sisters who have typical FMD, then you never know if an isolated aneurysm, like we're seeing in this third sister, or first sister really, is just an isolated aneurysm or whether it's related to fibromuscular dysplasia. Now, this is an example of arterial tortuosity, or the so-called S curve. You can see to the left is the head, to the right is the feet. The artery, the blood flow is coming up here, it's curving around, and then it's going up to the brain. So this is what we've termed the S curve. It's a novel morphological feature of FMD. It occurs in other diseases, Loeys-Dietz syndrome, Marfan syndrome. Many patients with other conditions have tortuosity, but this is a characteristic feature of fibromuscular dysplasia. This is the carotid artery of this particular ultrasound. And some even have loops in their arteries. Here you can see a loop in the carotid artery. So in 2012, we published the first paper on the first 447 patients. We now have well over 4,200 patients in the registry. I don't remember the exact number, but we learned a lot from these 447 patients, and we continue to learn about how these patients present and what their natural history is, how they do in the long term. Now, one of the papers that came out of the registry was this, dissection and aneurysm in patients with fibromuscular dysplasia, findings from the US registry. This was published in the Journal of the American College of Cardiology in 2016. And at this time, we had over 1,000 patients. 921 patients had complete aneurysm and dissection information. And these were the patients that were reported on. So in this series, 22% of the patients had an aneurysm somewhere. This is an example of aneurysms in the kidney. The renal artery is the most common place, followed by the carotid artery, and then the arteries inside of the brain, cerebral arteries. We did a series of our patients, 100 of our patients, and 35% of them had aneurysms. And I think the difference is in this study, not every single patient had head to pelvis imaging, But in the series we looked at, every patient had cross-sectional imaging from the head to the pelvis. And then dissection occurred in 26% of patients. The top figure is a carotid artery. You can see it's split here. The bottom figure is a vertebral artery, the artery that goes to the back part of the brain. And the most common place you get this is the carotid artery, followed by the vertebral artery, and then the renal artery, and guess what? The coronary arteries. We didn't know this in the beginning, but 10.5% of all the dissections when we published this paper were coronary artery dissections, which is now known as spontaneous coronary artery dissection, or SCAD. And here's an example of that. You can see the split in the artery where the white arrows are. And this is a catheter you put inside of the artery, and you can actually see the tear in the artery. And this is what's called the true lumen, and this is what's called the false lumen. And so these are examples of dissections in the arteries of the heart. You don't get the string of beads in the arteries of the heart, but you do get dissections. When you see an artery that's normal, and then you have a long, smooth area of narrowing, and then it's normal again, that's a dissection. So in this particular image, 2 of the coronary arteries were dissected. When I see SCAD, I classify them in one of 3 ways: SCAD alone, SCAD, and FMD. And my feeling is this is the same condition. It's the same disease. And SCAD with other extracardiac abnormalities, such as tortuosity, ectasia, aneurysms, and dissections in other vascular beds. So every patient with SCAD should have head to pelvis imaging. And in this series of patients, 41.7% of our patients had an aneurysm, a dissection, or both. So that's a pretty high number of aneurysms and dissections, and that's why it's so important to do cross-sectional imaging. We prefer CT angiography because the resolution of it is better than magnetic resonance angiography, but either one of these will do. We do CTA on all of our patients. I'm just finishing up here that there's been 3 studies published that has shown the same gene locus in 3 separate conditions. It's been shown first, Stephanie DeBette's article on PRACTA1 increases susceptibility of cervical artery or carotid artery dissections. Then there was a paper published in PLOS Genetics by Chiondo et al. That involved a lot of us showing that this is a susceptibility locus for fibromuscular dysplasia, PRACTA1. And then it's also a common genetic variant locus associated with spontaneous coronary artery dissection. So these things are related genetically in some form. And I'm just finishing with, this is part of our team. This is Jason, who's you're going to hear from next. He's really the brains of the whole organization. Valentina is the first author on the paper that you're going to hear about shortly. She handles all the fibroblasts in the study. This is Dr. Katie Dodoff, my partner, Annette King, my nurse practitioner, and one of our cardiology fellows, Catherine McAllis, who worked with us from a number of years ago. So with that, I'll stop and thank you for your attention, and I will turn this over to Jason.
Right. Thank you, Jeff. That was a great background and introduction. That was really helpful. Well, I'll say good morning to everybody. I'm from here in Sydney, Australia. I have a joint appointment in Sydney, Australia, and also at Mount Sinai School of Medicine in New York, where I was based full-time until about 2020. I still have a small lab in Mount Sinai, New York, and the work that we're going to present and speak about this morning has been the result of ongoing studies that are still going to this day. So it's a real thrill to be here, and I'd thank up front Pam Mace for what she does for FMD Thank all the patients and my patients for joining in, both from the States and from Australia. And I think particularly the patients that participated in this study. So we, when I was started at Mount Sinai, which is about 2009, FMD at that stage was unquestionably the most common cardiovascular disease about which we knew absolutely nothing whatsoever in terms of what was the cause, what was the genetics, what was underlying it from a biological and a pathological point of view. So in about 2010, we started really putting our thinking caps on and trying to figure out how could we actually make some serious progress in understanding what causes FMD. And right from the outset, it was, actually a historical point that physicians, pathologists, doctors, going back to the very first description of fibromuscular dysplasia in 1938, had appreciated that fibromuscular dysplasia likely involved this process of fibrosis and these fibroblast cells. So thinking about that and reflecting on that, after a while, we just thought, well, let's get fibroblast cells from patients and study them and understand them. So that led to what we call the Define FMD study, where we're actually able to get fibroblast cells from FMD patients and healthy controls. And as Pam was saying, there was 83 patients and 71 controls in this study. We've actually got over 400 that have done this now, where we actually take a small skin biopsy from the inner upper forearm, so from just up around here. And it's about a 3 millimeter punch biopsy, just local anesthetic. It's really straightforward and simple. It's the same thing you have done with a dermatologist when you're biopsying a mole or a freckle. We take that skin biopsy. That enables us to get these really small pieces of tissue that you can see there. We can then actually take them into the lab and take them into these culture conditions and actually grow them up in the lab. So what you can see here is let me just get this. What you can see here is what's called a tissue culture plate. We're actually able to put these pieces of skin down, put some growth medium on them, and they will actually grow in the lab and in the dish. This is Valentina de Escamarade, who's the first author on the paper we just published. This is Valentina handling these cells and looking after them in the tissue culture lab. So what you see here is what we get after about a month in culture. This at the bottom here is the original piece of skin. And here are growing out hundreds, thousands of these fibroblast cells. You can see here's one cell here. You can see there's hundreds that we're showing here, and they just grow out from this piece of skin. And this is actually a very standard technique. It's been done by many labs around the world. And we were able to use this technique to get these fibroblast cells. And then what we did was we can actually collect those cells off the tissue culture plate, harvest the genetic material, the RNA, and then actually sequence and process that RNA to look at the levels of almost 100,000 different genes that are expressed in those fibroblast cells. So this is called RNA sequencing. It's a very, very standard tool that we use in research, but enabled us to look at the gene levels in these fibroblast cells from patients versus controls. So we set about designing this study, and this is the Define FMD study. This is what we did. We recruited patients that had multifocal FMD documented by imaging with a consistent clinical history, and they were also enrolled in the US FMD registry that Jeff was speaking about. And then we, in parallel with that, enrolled controls that had an absence of FMD both on history and physical exam. And we matched those to the patients in the study. So we matched them by gender, by race and ethnicity, age, smoking, BMI, which is body mass index, or their how heavy they are, basically, the number of blood pressure medications. And we had an overwhelming response to this study. Both my mum and my wife are actually controls in this study. And this study is actually ongoing today. As I mentioned, we're still recruiting subjects into this at the moment. So what you can see here, this is Valentina again, who did all the cell work. This is what's called a tissue culture incubator. It looks a bit like a mini fridge or a bar fridge but it's kept at 37 degrees at body temperature. And these are all plates from patients and controls in the study. And we're growing their fibroblast cells here in this tissue culture incubator. Then we grow these cells for a couple of months to get off lots of fibroblasts that we can then use for our research studies. And just to say that we've now enrolled about 440 subjects into the study. It's roughly half FMD patients and controls. And Valentina has done an amazing job handling all of these cells and doing all the processing. And as well as the cells, we're also acquiring DNA and plasma and serum from blood and some other biologic materials we get from growing these cells. So one concept that I think is really important to explain to everybody now is that we now understand in biology that genes just don't operate in isolation. So when we have one gene, it's like throwing a rock into a pond, right? There's a ripple effect that goes out into that pond, and there's a spreading effect of genes and gene changes. So one thing that's actually evolved in the way we think about diseases is that we understand when there's a critical genetic change that can affect tens, hundreds, thousands even of other genes. And what we wanted to understand was, what are these big groups of genes that are moving and changing in fibromuscular dysplasia? And what are the critical genes amongst those that control them? So this is called a gene network. You can also think of gene networks a bit like a pyramid, and that we think about a top gene at the top of the pyramid. When we change that top gene at the top of the pyramid, it can affect the levels of many other genes. And there's a sort of cascading effect, or when you throw that rock into the pond, that ripple effect, that spreading out effect. So what we wanted to understand was, what are these groups or networks of genes that are involved in FMD? And then once we found those networks of genes, what are the top apex or key driver genes that control these networks that cause FMD? That was the goal of our study. So I'm not going to explain any of this, but I just want to highlight there was some super complex computational biology, systems analysis, genetics that went on through this study. What we did, we took all females, 83 cases with very clearly defined FMD, and 71 controls that didn't have FMD. We did our skin punch biopsy and grew fibroblasts. We also took blood, and from the blood we extracted DNA. And then we took that information, took these cells, and we applied this really complicated but quite cutting-edge pipeline where we basically identified these networks of genes that all work together to cause FMD. And then we tried to figure out how that most important network caused FMD. So I have to give a lot of credit to the bioinformaticians, the statisticians that were involved in this project, and Angela Ma did an amazing job in handling all of this really complicated bioinformatics and statistical analysis. So what do we find? This is a network we found of genes using those patient-derived fibroblasts that was strongly associated with FMD. And what you can see here, it's a bit like a hub and spoke thing, where you've got these genes that are in this central hub that are controlling the levels of hundreds of other genes spread around the periphery. So this is a network of genes that we found really, really strongly linked to FMD. We actually found about 25 different networks, but this particular network was the one that stood out as being really, really strongly linked to FMD. And then we identified that within this network, the network had about 775 genes in it in total. You can see there's lots of these little genes out the side here. We found that within this network, there were 14 genes that seemed to work as those apex genes that could control the rest of the network and have downstream effects. And within these 14 top key drivers, you can see those 14 are shown up here. There's 10 of them there and 4 around the edges. Within those genes, we became most interested in this particular gene that was UBR4. And there's a few technical reasons why we became interested in UBR4 that I won't go into. But we decided to focus on UBR4 to understand what this network of genes was doing to cause FMD. So what we did was we created, this took about 2 or 3 years to do, but we created a mouse where we actually knocked out or removed that gene UBR4 from the arterial wall of the mouse. And what you see here is this is actually a cross section of the aorta of the mouse, and shown in red is the UBR4. So this is a normal mouse, and here's our mouse where we've removed UBR4 from most of the cells in the vessel wall. You can see there's still a tiny bit of red. It's not a perfect system, but there's dramatically less red or UBR4 between the normal healthy mice and the mice where we've removed UBR4 from the vessel wall cells. And just for the physicians and doctors, I can say we specifically took out UBR4 in smooth muscle cells. And we did a lot of work to link smooth muscle cells to fibroblasts and make that connection from the patient fibroblast cells to smooth muscle cells in the mouse model. So we made these mice, we removed this UBR4 gene as our way to study that network of genes. And what did we find? We found that mice where we'd removed UBR4 from the arterial cells have dramatically larger aortas. This is the abdominal aorta. Here you can see the normal mouse. This is about 2 millimeters across. Here in the mouse where we've removed UBR4, there's a dramatically larger aorta. It was about 40% larger in diameter than compared to the normal healthy mouse. This is actually showing the thoracic aorta, and the heart is actually sitting down here. This is in the normal mouse. This is what's called the ascending aorta. This is the arch of the aorta, and this is where the descending aorta comes. So this is all up in the chest, and this is one of the carotid arteries coming off here. But you can see here in the mice where we've removed UBR4, the aorta is visibly larger than in the normal healthy mice. So what was really interesting to us, as Jeff said in his first presentation, that we know that FMD is associated with aneurysm, with dilation of arteries, with ectasia. And around the same time as we were starting to get our mouse data, we actually saw this paper that involved Esther Kim and Brian Wells that actually had clearly demonstrated that another of the features of FMD is that FMD patients do also tend to have larger aortic diameters, just as we'd observed in our mouse model, as well as these aneurysms you can see here in this renal artery. So it actually matched up one of the key features of FMD that we now understand had perhaps not been appreciated over the decades, but is actually one of the defining features of the disease, along with aneurysm, dilation, and ectasia, as Jeff was saying. So to summarize what the study found, and I've only presented to you really the highlights and the key elements, but we found this gene network, this cluster of 775 genes that's strongly associated with fibromuscular dysplasia. And we found and validated that this gene UBR4 can control that network. When we removed UBR4, that gene, from cells in the wall of arteries, mice developed some of the features of FMD, which particularly includes this arterial dilation aneurysm phenotype. And in other experiments that I haven't shown you here, we showed that those enlarged arteries were mostly because of changes in collagen and matrix. Now, they were particularly complicated experiments, so I haven't shown you here, but when we looked at the changes in the network, it was consistently changes in collagen and matrix that appeared to be involved. Collagen and matrix has actually come up in several of our other genetic studies, some of those ones that Jeff was talking about before with Practa1 and some others. Then collagen and matrix is one of the consistent themes we're seeing as what is causative for FMD. So what is collagen and matrix, and what has our study shown? Collagen is a protein that provides structural support to tissues. And you can see here in this image This is the surface of a tissue like the artery or the skin or the gut. And here are some cells, and amongst these cells are collagen proteins that provide structural support, and also matrix, which are proteins and molecules that surround and support cells and tissues. So a great example of collagen is when you have your skin and you do that to your skin, you pick it up and it relaxes back down. That's collagen that can cause the skin to come back down. So you can imagine if there are changes in collagen proteins or in matrix proteins, it could easily be a setup or a cause for fibrosis, for dissection, for aneurysm, for dilation, for all of these features that we understand is fibromuscular dysplasia. So I just need to finish by thanking everybody. This is the front page of the paper that we published. The technical term was Integrative Gene Regulatory Network Analysis Discloses Key Driver Genes of Fibromuscular Dysplasia. Valentina, who I've shown you, was the first author and did just an amazing amount of work to grow all of those fibroblast cells, harvest them, and do a lot of the mouse work and other studies that were involved. Daniela, who was critical in seeing all the FMD patients along with Jeff Olin, and did a lot of help with enrolling all the subjects. Li Jiang Ma, or Angela, who did all the most of the bioinformatics for the study, and also Annette here, who helped enroll many of the patients. I'd shout out to Yang, who's from my lab, who did a lot of work in the post-processing and to help make the figures. A whole lot of people that contributed all sorts of different skills and techniques. Jeff, who was really important for helping set this up well over a decade ago, and many others that contributed so much. And just to share with you something that really gave us a thrill, the journal where this manuscript was published in, Nature Cardiovascular Research, chose to feature our article on the cover. And what they've depicted here, this is the sort of head of a mouse, this is the head of a human, sort of showing this network biology connecting things. It is a bit of it's a nice thing to have the cover shot of this journal with the heading Understanding Fibromuscular Dysplasia featured there. So thanks to the funders of the study, which was NIH, and also some very generous philanthropic donors who helped us do a lot of this work. So I'm going to end my talk there, but I think we'll open it up with Pam and Jeff for some questions. Thank you.
Great, Jason, that was terrific. I just want to share my screen again for a minute to show all the people on what actually is involved in this. Jason explained this in the most simplistic terms possible. So let me just see if I can, okay. Can you see that, Jason? Is that showing?
Yeah, I left that slide out, Jeff.
These are just one of the many figures that were involved in a paper like this. I mean, look at this. It took years to get this mouse model to the way we wanted it, but all of this underwent very intensive review in one of the top journals in the world, and required a lot of, here's the figure that you saw, but you see, it's a lot more complicated than that. And to get to that point took a lot of work, years of work, before before we really started with this particular part of the study. So I just want to stop there so everybody can appreciate that this is not just a simple little thing, but when you talk to the computational people and the mathematicians involved, it's hard to even talk to them to understand what they're talking about. So
yeah, it's
anyway,
taken very long, time to get to this point. So it is very exciting, and I don't think that I thought it would happen in my lifetime. And like I said, we started this, over 2 decades ago. And so it's really exciting to be at this point. And we have all of you guys to thank for that, and researchers around the world. We have a lot of physicians on the Zoom tonight, which I appreciate them being here and all of their support. So I just want to thank everyone. We've got patients literally from all over the world on this call. So I'm going to go ahead and start with some of the questions. So someone asked, is there going to be a blood test so that family members can be affected?
Yeah,
Or-
thanks.
Okay, that's
Is there going to be a
Be
blood test?
tested.
So in fact, we've published a paper back in 2020 where we did exactly that. We looked at blood and specifically blood proteins to see if we could identify a set of proteins, a set of markers in blood that was unique to FMD. And we were actually successful in doing that. And we showed in a research paper, at least, that it would likely be possible to develop a blood test for FMD. The challenge was just funding, and we tried a couple of times to get grant funding for that project to roll it out and see if we could develop it into a clinical tool. But we were actually unsuccessful in getting research funding for developing a test. And I think it's just the reality of, getting funding to do research, because this particular study that we just showed made the first mouse model. It understood I think some of the core aspects of the disease, and it perhaps opens up the possibility, at least, of thinking about therapies to reverse the process. It's generally a lot easier to get funding for something that might have those opportunities, and particularly developing a therapy rather than a blood test. And I think some of the comments from the reviewers was, well, you can kind of screen for FMD with ultrasound, with MRI, with CT. Would a blood test really change things that much? So I think it's probably possible, but we unfortunately haven't been able to proceed with that work.
Thank you. So someone asked about participating in the research, and they actually live in Canada, but how someone would be able to participate in the research at Mount Sinai.
I already answered that.
Yeah, some
The
people,
group
I know a couple people had trouble signing on at the beginning, Jeff, so they
No,
probably
I
missed
just answered it by writing. Yes, you can participate.
Okay,
Yeah.
all right.
It went out to everybody.
All right, well, I'm busy
Worth
reading.
saying that because we need to get the skin biopsy, you'd need to come to New York, to the team in New York, to do that. So it's you have to come to New York to participate, if that's possible.
Yeah, okay. And okay, this is a patient who has FMD. She's had 2 scads. Her daughter was incidentally diagnosed after a headache, and she has 2 other daughters. Should they be tested?
Jeff?
About somewhere between 10 and 20% of first degree relatives on the female side inherit this. Inheriting it and the genetics of it are 2 separate things. So I leave it up to the patient, and I ask a few simple questions. First of all, how old are we talking about? If the person is less than 25 years old, I say wait, because I've seen 2 cases where we screened people early and missed it, and then 10 years later we saw the findings of fibromuscular dysplasia. So I'd wait at least till age 25. And then I ask, do they have migraine headaches? If the answer is yes, that would make me recommend screening much more. Do they have high blood pressure? Do they have what's called pulsatile tinnitus, or a whooshing, swishing sound in their ear? Any of those 3 things, they absolutely should be screened. And then for all the others, I say it's up to the individual family, and what tests are you going to do to screen for it? We do ultrasounds here, but not everyone can do ultrasounds and screen for FMD, and we probably miss some cases by doing ultrasounds. So I leave it up to the family members, and together we have shared decision making, which is a big thing in medicine now, and we come up with a plan. If the parents or the brother or sister really want to screen their children, sure. I tell them just wait till they're old enough, but we make the decision jointly.
Thank you. So Jason, someone asks, do we have to worry about our aorta? And they stated that then is FMD caused by a lack of UBR4
Mhm.
and a lack of collagen or an excess of collagen?
Mhm. So, I mean, first to the aorta question. Aortic dissection, and Jeff, you comment please, but aortic dissection can occur in FMD, but it's pretty uncommon as part of FMD. And I haven't seen it. Jeff, how many aortic dissections have you seen in FMD?
Yeah, we've seen a few, but aortic aneurysms definitely occur in FMD. But
Right.
since every patient gets head to pelvis screening, you're going to pick that up. So you should not worry about it as
Yeah.
long as you've been evaluated properly.
Exactly. And so the other question about UBR4 and causing FMD and so on. Yeah, we've been thinking a lot about that. We've known UBR4 in these results for about 2 or 3 years now. So we know when we push this, when we reduce the levels of UBR4, when we knock it out, we cause certain changes in that network that lead to the mouse looking like it's got aspects of FMD. So we have been working on ways we could try to push the network back in a favorable direction to try and favorably reverse some of those changes we saw in the mouse. And that's been ongoing work in our lab now for well over a couple of years. It's technically, challenging, as was this study, but it's certainly where we're pushing towards, with a view one day, and it's not going to be anytime soon, but one day maybe seeing if we could develop a therapy.
Thank you. Okay, so someone's asking, there's a lot of questions coming through, about targeted therapy for UBR4.
Right. Yeah, that's a great question. So I think what's really cool about this study is until now, and there's been a couple of other genetic studies. I should shout out to my colleagues, Santhi Ganesh, Nabila Bhutanaji, and Mark Lindsay have done great things on helping to define the basis of FMD and others. So it's not just this study. This isn't truly the first study, but I think it's an important one. But the first step in identifying treatments is the targets. And I think now we've got a number of targets. It includes UBR4. It importantly includes that whole network that I showed, plus a couple of others we've found in some other studies. So absolutely, we're looking hard at UBR4, but particularly also the network as well, to see if we could push the network back in a favorable direction to change collagen and matrix. And I'd also say that it's not just FMD that involves collagen and matrix. Collagen and matrix lies as the cause of many, many different related vascular disorders that includes SCAD, as Jeff was saying, cervical artery dissection, but even other ones that are not closely related to FMD, like Marfan's, vascular endless loss. They're all collagen matrix problems. So it does make a lot of sense to be focusing on collagen matrix and that network that I showed you.
Thank you. So someone asks, will there be a cure and should we be taking collagen?
Yeah, so the answer to the collagen question, have no idea. I don't necessarily think it does any harm, but there's no data to show it helps. So I don't have a strong view on should we be taking collagen. I think a cure for FMD is a long way off. And yes, we're making baby steps towards some kind of therapy or treatment, but that's going to be minimum, 10 to 15 years of work and lots of hard research. So nobody should have their hopes up for any types of cures or treatments in the short to even intermediate term. I think one of the other things we'll need to think about with any treatments for FMD is that it may need to be given long-term. And for long-term therapies, we need to be really, really sure about safety. So, if we're going to give a drug for a decade, 20 years, it's going to have to be super safe. And that's going to take a lot of very, very careful clinical trial work. So we've got a lot of work ahead of us and it's going to take a lot of time, but we're on the journey.
Thank you. Okay, someone's asking about the progression of FMD and for the people that they think it progresses in, do you think that it's an environmental factor or a genetic factor? I know we typically don't think FMD progresses.
when you talk about progression of disease, there are two aspects you should consider. One is if you do careful imaging and you see the string of beads in certain arteries, later on down the line, you will not see beading in other arteries. In other words, that aspect of it does not progress. And most people who present and are evaluated because they've already had an event, such as a dissection or a tear in the artery or an aneurysm, and that's how the FMD is discovered. So patients who are diagnosed with FMD incidentally, when they get imaging for some other reason, often have no bad things happen to them. Many people live their entire life without any events, and they have FMD. And the events I'm talking about are stroke, heart attack, dissection, and aneurysms. And like I said, they usually are the presenting feature which allows you to diagnose FMD. It still could occur, but it's very unusual. So I wouldn't think of progression like you're going to have beating all over your body in 10 years. That doesn't happen.
Jeff, do you want to explain carotid webs?
Yeah, I will. The last person also said, is it genetic or environmental? It's probably a combination of multiple things. Like, why do 70% of patients with FMD not have a dissection, but 30% do? I mean, there's a number of other features besides one gene that's causing this. Carotid webs are something that occurs in the beginning part of the internal carotid artery, right where the carotid artery divides into the internal and the external. And it looks like a web. It's a little indentation there, and it's associated with a very high incidence of stroke. We don't know why that is. So many people recommend if you see a carotid web that you, at the very least, go on antiplatelet therapy like aspirin and clopidogrel. But many people also suggest that you should consider stenting or surgical correction so you don't get a stroke. We don't know what causes it. I don't think it's part of the FMD that we're talking about. The original papers on carotid webs were fibromuscular dysplasia in Afro-Caribbeans, and they were talking about carotid webs. But the more I read about it, the less likely I think these two conditions are associated.
Okay, thank you. And Jeff, someone's asking how to contact your office to participate in the research.
Type my name into Google and you'll see my office number.
Okay, it's also on our website. So under the registry, you can also find it on there. And
if I could just make a plea for everyone out there, Pam has run this registry since we started it. And we've been doing this on a shoestring budget. We desperately need funding for the registry. We are so underfunded that the registry is at risk of not being able to continue. So whatever anyone can give to help continue the registry would be really helpful. We're talking about much less money than it takes to do the kind of experiments that Jason explained to you. That takes millions of dollars. We're not talking about millions of dollars here. So please consider donating whatever you can afford to donate so we can keep this going.
Thanks, Jeff. You
Okay,
know, the
Pam.
registry, we started the registry years ago, and it really was the catalyst for research around the world. Once the findings started being published, more and more researchers got involved and were contacting us, and everyone started working together. And the problem is, you know, the funding's just not there. So this is the worst year that we've had in over a decade, and we're already down at least $50,000 this year. There is no office help. And so it's getting harder and harder to run the organization. And so our future is kind of uncertain at this point. So we're hoping that, you know, year end, the donations will come in and that we can keep the registry funded. So I appreciate that, Jeff. Okay, so what do you think the role of estrogen is, and why more women are affected?
We have some experts on the who are listening to this. Marissa Tweet, who's done an incredible amount of work with SCAD. Esther Kim is on. Jason, you want to take the estrogen question?
Sure.
Or
Look, I think hormones are likely to be involved in FMD, SCAD, and arterial dissection, but I think it's more complicated than we appreciate. So I can share with you some other data. So Jeff showed in his slides that there's another gene, PRACTA1, which was the first gene we ever found was linked to FMD. This was in separate studies, and PRACTA1 is also implicated in SCAD and cervical artery dissection. So we've made a mouse where we've taken PRACTA1 out, so we knocked out the gene. So the mouse doesn't have the PRACTA1 gene. And we've looked at the vessels of those mice. And we see very clear sex differences in the vessels of those mice. So the female mice have different contraction, relaxation, and other functional changes compared to the male mice. And that change in those aortas is completely independent of hormones. So I think the hormones are involved, and I think a great example of that is when we see peripartum SCAD. So spontaneous coronary artery dissection that happens around the time of childbirth, usually a week or two before versus several months after. That, I think, is very likely due to the hormonal changes of pregnancy. But I think there are also changes going on in FMD and SCAD, cervical artery dissection, that are unrelated to hormones. And understanding all of that is going to take us quite a bit of time and effort. We are working on that. It's, as I said, that's one of the key things we're doing. Now, that's actually happening here at the Victor Chang Cardiac Research Institute in Sydney, Australia. But I think it's quite complicated. And I think as we know, there's some women that will have arterial dissections in their 30s or 40s when they're clearly premenopausal. There's a group of women that get into the perimenopausal period and start having arterial dissections. And then there's people that have arterial dissections when they're actually we've gone through menopause. So it can happen at these different phases. So I think there's a lot we still don't understand, but hormones are linked at some level.
Okay, and can you talk a little bit about men in FMD?
Yeah, so
if I
Jeff,
could
go
just
ahead.
comment about the estrogen thing. Many patients, postmenopausal patients, ask if they can take estrogen and is it safe? And we don't really have a definite answer, but what I always say is it would be good if you were taking it, not to take estrogen pills, but maybe consider the patch or even more localized estrogen. And there may be a slightly increased risk of dissection, but we don't really know. So if they want to wear an estrogen patch, that's fine with me. I don't know if the other experts who are listening to this feel the same way, but that's what I tell my patients. Sorry to interrupt, Pam. You
No,
were
that's okay.
mentioning men.
No, men in FMD.
Yeah, I think men in FMD is a great question, and men in arterial dissection is a great question. We have just because FMD overwhelmingly affects women, we have far fewer men in any of our studies, the registry and our Mount Sinai study. So it does happen. My sense is it has many of the features, but it's not exactly the same in men. I think they present slightly differently. The ones I've seen often have arterial dissection. I'm sure you've got some comments on this, Jeff, but I presume that there may be stronger genetic factors at play in men because there's obviously not the same hormonal changes that are probably causal to some extent in women. But it's a great question, but because of the fact that we see fewer men with FMD and SCAD, we just have fewer of them to study and understand. And indeed, the study I presented was entirely women. We actually didn't put any men into the study because we think maybe some of the drivers of FMD might be slightly different, and we just wanted to focus on a very specific population that likely had the same biology going on to find that gene network.
Thank you.
Yeah, one thing I'd like to bring up is that 50% of the patients who are referred to me with a diagnosis of FMD do not have it. So that's important because there are mimickers of FMD. For example, vascular Ehlers-Danlos syndrome. It's a disease, it's a terrible disease to have where patients dissect and have aneurysms rupture, and it leads to early death. And it's a genetic disease, and the gene has been identified, COL3A1. Well, I've seen a number of patients with vascular EDS who came to me because they were diagnosed with FMD. Because sometimes after a dissection, an artery during its remodeling can look like there's a little bit of beating present. So I would just make sure that if you've been diagnosed with FMD, that it's the real thing, and that things that can mimic it have been excluded.
Thank you. So we're getting a lot of questions about participating in research. And so for the people that can't make it to Mount Sinai, Dr. Ganesh, if you look up Ganesh Lab, there is a section on there you can go to for patients, and you can participate from anywhere. And they've been actively recruiting patients, but they work with your physicians back home wherever you're located. So you would just look up Ganesh, Ganesh Lab, and the information will be there. And so, Jeff, somebody asks, Jason, aren't you able to send out kits to patients like Dr. Ganesh does?
Yeah, unfortunately,
No.
because we do the skin biopsy, that
Right.
requires local anesthetic and it's someone really skilled to actually do that. You can't do that yourself. So yeah, unfortunately, you do need to come to Mount Sinai to do it because you actually need to process that skin sample quite quickly so that it's, it's healthy and we can get all the cells to grow out of it. So it's just the reality, the nature and the design of the study.
Not only that, but we want to make sure the patient has FMD.
You're right.
So I'm just not going to go by what the outside records show without looking at the images and examining the patient and talking to them. That's the only way they get into this study.
Right. Yeah, it would affect the accuracy of the data. So someone is asking, are you born with FMD? And do you know the percentage of children, if a patient has FMD, the percentage of their children that actually have FMD?
Yeah. There is a form of FMD where it occurs very, very early, like when you're 6 months or 3 months. It's got a very malignant course. It doesn't behave anything like adult FMD. But I've taken care of 6-month-olds who have had dissections of their mesenteric vessels, their intestines, and various other organs. Then there are children who aren't quite that young, but have FMD, at age 4 or 6 or 8. And I have one patient whose mother has one type of FMD and daughter has another type of FMD. So yes, it can occur in children. We don't know how often. Kevin Myers at CHOP, Children's Hospital of Philadelphia, is part of our registry for pediatric FMD. We know very, very little about that.
Chloroquine antibiotics and FMD. Oh, and are you born with FMD? I don't think you answered that.
Which one?
Are you born with FMD? And then can you take chloroquine antibiotics with FMD?
Fluoroquinolones. We don't know if you're born with it, but if somebody has it at 3 months, you probably are born with it. But the kind that you all have who are on this call, you're not born with it. Something happens after puberty. We don't know what it is, but fluoroquinolones like ciprofloxacin and Levaquin and things like that have been associated with rupture of tendons, and it's been associated with dissections. And there have been several papers published that if you've had dissections, you probably should not take a fluoroquinolone. I don't know how you feel about it, Jason.
Yeah, no, I often will pretty routinely tell my patients, not in the first visit because it's complicated, but tell them to just consider themselves allergic to the fluoroquinolone antibiotic. So that's ciprofloxacin, gadafloxacin, there's a whole bunch of them. I do think it's, that's not an antibiotic class that's used that often. There's plenty of alternatives. So I think they're fairly easily avoided. So I just tell all my SCAD FMD dissection patients just to think of themselves as allergic to that. And I write it out on a piece of paper so that they know about it. Pam, I was happy to share just one quick slide, I think, about the question of are you born with it and what's the genetics. I hope this is helpful to people.
Sure. Yeah,
Just
there's
to
a lot of
show,
questions.
yeah, just to show this. So there are two broad types of genetic inheritance we think about, and these are common gene changes and what are thought of as sort of rare variants. And I think this is a really important concept to convey. So we now understand that most of FMD, most of SCAD is due to common gene changes. And in that network that I was presenting, the UBR4, the PRACTA1 that we've been speaking about, these are all related to common gene changes. So most common cardiovascular diseases and many diseases are actually these related to common gene changes. So most coronary artery disease, most diabetes, most high blood pressure, height, weight, most arthritis, most stroke. So when people are thinking about, are you born with it? You do know that most high blood pressure, this is not related to FMD, it does tend to run in families, but there'll be some family members that miss it, some that skip it. It's a little bit random. And that's pretty much the inheritance pattern of most FMD and SCAD. So to some extent, people are definitely born with these genetic changes. And these genes involve, there are sort of literally hundreds of genes potentially involved. We've identified a bunch of them now for FMD and SCAD, but there's probably many, many more to identify. The other type of genetic inheritance is what's called rare variants. And that's basically when there's a single gene change, and that single gene change alone can cause disease. And we've been speaking a little bit about vascular Ehlers-Danlos. That's an example of a rare variant disease where a single change in the gene collagen 3A1 causes a disease, or in Marfan's disease, a single gene change in fibrillin 1 causes Marfan's. There are probably some patients with FMD, and we've definitely identified some patients with SCAD that have a rare variant as causing their disease, but this appears to account for about 10 to 15% of people with SCAD, probably similar numbers in FMD. For 80, 85% of people with FMD and SCAD, it's going to be common genetic changes, but most likely, if you're going to have it, you'll be born with it. But whether or not you've got it at the moment, we don't have any genetic tests for this, and we can't actually do genetic screening formally for FMD or SCAD at the present time. So I hope that's helpful. It's complex, and it's both of these types of genetic changes can be involved.
But if you want to be optimistic, take a look at what's happening with sickle cell anemia. It is absolutely curable now with gene therapy. The problem is no insurance companies will pay for it, and you have to go through a long process to get it. But it's amazing. And in when I'm long gone from this field, gene editing and all that is going to be the way of the future.
Yeah, that's obviously where we're focusing our efforts now. But it's I don't want to get people's hopes up. It's a lot of work, but that's what we're working on at the moment.
Yeah. So someone emailed me, they thought they heard someone say that this is curable. Can you just go over that again?
That was Jeff.
It was a joke.
I just want to clarify it for people.
It was a patient who had a narrowing of a artery going to the kidney with really high blood pressure. And we did an angioplasty on her, and we cured her hypertension. She didn't have to take any more blood pressure medications. And she sent me a note and a vase saying, "Thank you for curing my FMD." That's all. I didn't cure her FMD, but I cured her hypertension.
Hypertension. Oh, okay. Thank you for that clarification. So there's a bunch of questions on exercise and FMD, and people are just told they haven't taken aspirin. Do you want to talk about you know, I know there's no umbrella statement for exercising. It depends on what arteries are affected and the severity of your disease, but and then kind of maybe just some guidelines on what, you know, monitoring. And I think a great paper that if the patients aren't familiar with it is the International Consensus paper on fibromuscular dysplasia. It's a free paper. It's Sage Journals, and I think you should definitely print that out and save it.
Marissa Tweet, who I saw on, is listening, and Sharon Hayes from the Mayo Clinic, and I published a paper in European Heart Journal on exercise after SCAD, so people can take a look at that. But with FMD patients, if you just have plain old FMD and nothing bad has ever happened, and your blood pressure is not really, really high, it's well controlled, you pretty much can do whatever you want. But if you have it in the, I try to tell every patient, don't let anybody do anything with your head or neck. No chiropractors, and don't let anybody do a deep tissue massage where they're pulling on your head or twisting it in extreme positions, because that can be associated with dissections. That's what we would worry about with that. If you've already had a dissection, it's a different story. We would limit weightlifting to a certain amount. They have to wait a certain amount of time after the dissection before they even do that. We don't really have time to go into what the whole program would be. And certainly everybody doesn't do it the same way. But if you have an aneurysm or a dissection, exercise programs need to be spelled out very carefully for the patient. If you just have FMD, do whatever you want, but don't let anybody touch your head.
Okay. How often should a patient have head to pelvis imaging?
One time.
Okay.
But if I see somebody in their 30s and they have anybody in their family who's had a cerebral aneurysm, then I repeat an MRA of the head every 10 years. There's no data to support that, but.
Okay. So there's someone in Australia who wants to see you, Jason. I've been diagnosed with FMD. Do I need to see Jason or just see my local vascular doctor?
Look, it's up to the patient. Up to the patient. If as Jeff said, if they've got simple FMD, it's up to them. I mean, I'm really easy to find on the internet. There's not many Kovacic's, Jason Kovacic's in Sydney, so I'm easy to track down. But yeah, I've seen patients from all over, and happy to do so.
Okay. Do you guys, are you good? A few more questions? It's about 8:10.
Yeah, a couple of questions are good, Pam.
Okay. All right. Perfect. So migraines and FMD, are they caused by the FMD?
Well, they're associated with FMD. Whether FMD caused the migraines or not, we don't know. But there is a very strong association in the registry. About 65% of patients have headaches, most of which are migraine type headaches. There are a lot of new drugs now to treat migraines that weren't available before. Many people would take a triptan for their migraines. We advise against that because of some papers that have been written about increasing the rate of dissections, coronary dissections especially. And there are so many other drugs that can be used, but there's an association. We don't know if it causes it or not.
Okay.
Maybe I can just add, Pam, we also know that some of the genetics of migraine is shared with FMD and SCAD. So the PRACTA1 gene is also a gene that causes migraine. And at the end of the day, they're both vascular disorders. So migraine's a disorder of vascular regulation and tone in the brain. And FMD is obviously a vascular problem. So, you know, I agree with everything Jeff said as well.
And so there's a couple questions on the relationship with FMD and Ehlers-Danlos. And then someone also asked, is there a relationship with MALS celiac compression and FMD?
I'll take the last one first. Median arcuate ligament compression is very, very, very common. I run the vascular lab at Mount Sinai, and we see it all the time. And 99.9% of the time, it doesn't cause any symptoms, and it's not dangerous, and it's not associated with FMD. In the early literature, there used to be written that there is an association of vascular Ehlers-Danlos with FMD. But that has not been borne out. And the reason I think people thought there was a relationship is that Ehlers-Danlos patients dissect, and then their arteries remodel, and it can look like FMD, but Ehlers-Danlos, vascular EDS, is a bad disease. There's no cure for it right now, and there's no real treatment for it, but it is not associated with FMD.
Okay. Jason, do you plan on doing any studies with males? So I think there's an interest on here.
Yeah, absolutely. So yeah, males is definitely on our radar. I didn't show it, but in the study I presented at the beginning, we looked both in female mice and in male mice, and we did them separately, and they both looked exactly the same. They both had that same aortic dilation. So in that mouse study with that gene network and UBR4, it appeared to affect both sexes equally. We're working on PRACTA-1, and we're absolutely, in that case, we're seeing differences between male and female mice. So I think that's being a very interesting study. And I think, collectively, probably something that we will do with Santhi Ganesh and Nabila in Paris and others, when we pool our samples and we can combine power to get enough males, it's very logical to look in males to see what the causes are and what we can figure out in males, because I think males are a little bit different, a little bit unique, but it's still quite likely that whatever we learn in males will also inform us very broadly about FMD and SCAD in both sexes. Just as what we're learning in females, we're understanding in males as well. So I think it's a really important area of research. And although it's harder, just because we don't have the same numbers of subjects, it's something we really are working towards.
Okay, thank you. Should patients have genetic testing done? And what would that be? What would you be looking for?
Yeah, that's a great question. And the answer is sometimes, but it's not the kind of genetic testing probably that most patients are thinking of. We don't look for this gene network. We don't look for UBR4. That's still very much a research tool at the moment. The people that I send for genetic testing, and Jeff, you may want to comment, but if people have had, usually 2 or 3 dissections, if there's a question about if it's truly a string of beads or not, or maybe it's a healed dissection, if there are other features of other connective tissue disorders, so perhaps they might have super hypermobile joints or other features, if there's a really strong family history, if there is pregnancy-associated SCAD, or if people are contemplating a pregnancy, those are the kind of people that are sort of exhibiting some higher risk features or unusual features. And those people should probably undergo traditional genetic screening and genetic counseling with a geneticist usually at a tertiary referral center like University of Michigan with Santhi, or at Mount Sinai with our team, to have a regular panel of genetics looked at for aortopathy and other genes. So that's what I do, Jeff.
Yeah, I mean, if there's something unusual about the FMD, somebody has spontaneous ecchymosis or bruising or other features, they have eyes that look a certain way, or their ears are low set, or they've had multiple dissections and extreme arterial tortuosity, I will send that for the aortic panel. It's about 28 to 30 genes. And then there's another 30 or 40 that is included in a connective tissue disease panel. But I tell everybody the same thing. There's a chance, I don't know whether it's 10% or a little less or a little more, but there's a chance a gene will show up and it will be of unknown significance. That makes a lot of people nervous. It's a gene, but they don't know if it's causative of disease or not causative of disease. There's not enough information, and that comes up frequently.
Okay, thank you. Esther Kim says congratulations to everyone. So just thought I'd share that. So treatment of dissections, when is a stent used?
It's hard to, it's really hard to answer that because it depends where you are, who's going to be putting the stent in. And if you have someone who is very experienced, a neurointerventionist, putting stents in people with unusual vascular diseases, then you'd be much more liberal in putting a stent in than if you have someone who has done one or two of these things. So if someone is continuing to have neurological symptoms from a dissection, a carotid dissection or vertebral, I put a stent in then. If someone has a large pseudoaneurysm, that's like a big piece of the artery is hanging off the main artery, I have a stent put in then. But I can't really cover all the times when you would definitely put a stent in. Many patients heal on their own. And many coronary dissections, most heal on their own. And you probably should avoid stenting coronary dissections if you can. I mean, if you have to, you have to. But if you don't, most of them will heal.
Thank you. Is it safe to take low-dose aspirin to prevent further issues with FMD?
It doesn't prevent dissections, just so everybody knows. We recommend low-dose aspirin for everyone with FMD. It's never been studied, but if any little particles attach to the webs inside the artery, aspirin could prevent that from forming a clot and breaking off. So that's why we use it. But it doesn't prevent dissections, but it might, if someone has a dissection, prevent thrombus or clot formation, or at least help to prevent that. So the result will not be quite as bad as it would be if you didn't take aspirin. Again, none of this is really studied in a formal way.
Okay. A couple people commenting their children have FMD. They were diagnosed very young. So Dr. Olin, they're asking, did I hear you correctly at the beginning saying that once screening is completed and FMD lesions are identified, it is unusual to identify subsequent lesions? I know you addressed this before.
Yeah, I think there's been, not only is it unusual, I don't think it happens. I think I've seen maybe one or two cases where it's claimed that they didn't have beading in this area, and then later on they had beading. But so yeah, if you've done a good imaging study, now if you do an MRA and you don't see beading in an artery, but then you do a CT angiogram and you see beading, that's not the same thing, because MRA will miss very mild beading, where CT angiography probably will not miss it. So I would say if you've had a CT angiogram of your chest, abdomen, and pelvis, and of your neck and head, that wherever you have beads, that's where you have beads. You're not going to see them develop anywhere else.
Okay, for a male, can you just address a dissection and weightlifting? Carotid dissection and lifting weights.
Well, I can tell you this. They've done a study with weightlifters where they put an arterial line in them. And these are power lifters, and their blood pressure at the time they're lifting weights goes up to 400. So I wouldn't want an FMD patient's blood pressure to go up to 400. I don't know. I don't think millions of people lift weights and they don't develop a dissection. If you have a dissection, though, I'd be very careful about what kind of weightlifting you do.
So Jason, this is someone who has SCAD, and so they're asking about the genes for SCAD and FMD.
Yeah, no, that's a great question. Thanks. So yeah, um, we've done genetic studies on both conditions, and Esther Kim, who's on, and Santhi Nabila, Dave Adlam, very similar investigators have been involved with those. And the genetics are not exactly the same, but we do see a significant amount of overlap between the genetics of SCAD and FMD. So that PRACTA1 gene we mentioned, there's also a couple of others. LRP1 is another off the top of my head. And there's a few more that are common to both diseases. So I think that fits with what we see clinically, that as if we take people that have come in with SCAD and if we screen them carefully for FMD, it's probably around 50% have got some form of FMD, even if it's mild. So there is a significant amount of overlap. So, you know, some of us think of them almost as a different manifestation of the same disease. I certainly do. And Jeff and I argue that backwards and forwards, and we debate that at conferences and things. But there is some commonality in the cause and the genetics underlying things.
Okay. And there are a ton of questions in here still about collagen. Can you
Yeah,
people are asking,
yeah.
should they take
Happy
it?
to
Should
go
they not
back.
take it? Yeah, does it
Yeah,
help?
yeah.
So,
Yeah, so the answer to that is we just don't have data on collagen supplements, FMD and SCAD. It's very clear that changes in collagen matrix, although we're just starting on this, but the changes in collagen matrix appear to be commonly come up in all of our genetic studies, and not just what I presented today, but in the other studies we've done both of SCAD and of FMD, collagen matrix changes come up. We've got no data on whether taking collagen supplements will be helpful or even could be harmful. We just don't know at this time. I think it's a great thing that we've actually got to this point though, and we're starting to drill down on what the cause is. And I think changes in collagen matrix makes a lot of biological sense. But unfortunately, we just don't have the answers to those questions yet.
okay. And I think we're just going to take a couple more questions and wrap it up. I appreciate you guys taking this time. And so someone is asking, will there be gene therapy in our lifetime? I'm not sure how old they are.
Yeah.
Average age of diagnosis is
Important.
52. So let's go with
Important
that.
point. Yeah.
For FMD or gene therapy for anything?
No, for FMD.
Look, I think it's unlikely there'll be just a general treatment for FMD as a gene therapy. I don't think so. Could there be? I think as Jeff was saying before, one of the risks with FMD and SCAD and other dissection events is just recurrent dissection, because that's the thing that happens out of the blue, and there's a risk of recurrence. So once you have a dissection, a proportion will have another recurrence. Could we have a gene therapy to perhaps just target those people at risk of dissection or recurrent dissection, that we wouldn't have to give the therapy for decades? We could maybe just give it for a couple of months to stabilize them. In the next 10, 20 years, maybe that's something worth sort of thinking about and working on. But I don't think there'll be just a general preventive therapy that will give for decades to people, but some kind of targeted and focused therapy to reduce recurrent dissections or something like that in people that are having real trouble. That's something we're thinking about, but it's going to take a lot of work, a lot of resources, and a lot of time.
Okay. Well, I thank you both for being here this evening. I don't know if there's any other messages you'd like to share. I think that for me personally, I have a lot of hope for the future, and this was a day I never thought I would see. And patients do very well. And so actually before the call this evening, a gentleman called me and his wife was just diagnosed incidentally at the same hospital somebody called me from yesterday. And so it's the diagnosis rate is just, it's amazing. And so we appreciate everything that you guys are doing.
Yeah, well, we appreciate, what you're doing, Pam, through FMDSA, and I think it just plays such an important role for FMD patients. And, we're just pleased to play our part in helping treat the patients and helping try to find these causes of disease. So congratulations to you for everything you've done, because I think it's because of all of your raising of awareness and bringing the community together that we're in this place as well. So thank you.
I
Thank you very
second
much.
that. Pam is, I report to Pam. I do whatever she tells me to do, so.
We all do.
Okay, at least one of us knows that's not true. All right, well, I thank you. And for the patients still on the call, in case you have more questions, we do hope to, we're going to review the recording to see if it needs edited at all, and it will be shared in a couple days as long as the doctors agree to it.
Sure.
And then Dr. Heather Gornick and Dr. Santi Ganesh are hosting, this was planned several months ago, but on November 19th. And so she will be discussing genetics again, in case you walk away from this with a lot of questions. And that is going to be hosted by University Hospital. So you can contact us. Someone asked about getting our newsletter. You can go to our website and become a member, and then you'll get our newsletter with constant updates. So we appreciate everyone being on the call today. And Jeff and Jason, thank you so much for being here and for your expertise.
Thank you.
Thanks. Good night, everyone.
Good night,
Good
everyone.
morning, Jay.